Thank you, George, and welcome for all to our inaugural event. We're about to start the second year of the Nano Lecture Series. Oh, sorry about that. Yeah, that one. Yeah. Yeah, you can see the upcoming events for the whole academic year. We have, I think, 6 scheduled lectures. And the main goal of this Nano Lecture Series, which is part of the NEET Consortium, the NEET Consortium is the National Health Implications Research Consortium established by NIHS last year, and our Nano Safety Center is part of that consortium, along with other 8 research groups around the US. And the main focus of this consortium is understanding the fundamental nano-bio interactions of nanomaterials. So in this lecture series, we're trying to bring together applications and implications. So you'll see a good mix here. We have some presentations from the consortium members, which are focusing primarily on the nanotoxicology, nanosafety aspect, but also we have presentations from leaders in the field of nanotechnology. So those are the people that they're making new materials, new applications, etc. So we're also trying to cover not only human health implications, but also some environmental aspects of nanomaterials. So we are honored and happy to have Wenwan Zhong from UC Riverside with us today. I'll keep the introduction short. She's part of the consortium, and she's leading actually the corona characterization method development aspect of the consortium. So I think that's the only project funded currently by NIHS on developing novel methods for corona characterization. And corona is very fundamental and important for the nanocellular interactions. We do know there's a lot of evidence that the coronas, they make, they determine the bioactivity and the uptake at the cellular level, etc. Of course, it's a kind of a chicken and the egg. The nanomaterials also define the properties of the nanomaterials will define the corona. So how to measure the corona, especially how to develop methods that we can use in screening approaches for engineered nanomaterials, that's not trivial. So that's where Wenwan comes in with her group. She's leading this field for quite some time, and she's developing novel analytical methods for the corona characterization. So not only protein coronas, but also all sorts of coronas. So I will keep my introduction short. I mean, you can find everything about Wenwan on the web. So thank you for taking the time and visiting us, and look forward to your presentation.
Oh, okay. Yeah. Thank you very much, Phil, for your nice introduction and kind invitation. And also thank you all for coming here today for my talk. My group actually does a lot of work on studying nanoparticle-protein interaction and also characterizing protein corona. But my group is mainly a chemistry group. So our function here, I think, is to develop analytical methods and advanced analytical methods to help us improve the understanding of the nano-bio interface. So that's why my topic is here is actually how we apply and also develop advanced analytical tools to study protein nanoparticle interaction. So why protein absorption is of interest? It's because for nanomaterial now we see a lot of them like in our normal life and interact with them actually continuously, and people also use them in a lot of areas, including biomedical research. And then for nanomaterials, they have very large surface activity, so which will induce a very progressive and selective absorption of biomolecules like proteins on their surface. And then these absorbed proteins will serve as the interface when the nanoparticle interact with with biosystem, because the cells no longer see the particles as themselves. I didn't see the cursor. Yeah. Oh, okay. This doesn't work, huh? Okay. Yeah. Uh, anyway, yeah. So for a particle that stop in proteins, then this proteins layer will be the direct interface between the particle and the cells or other biosystems. So then that's why understanding the formation of this protein layer or atom biomolecule absorption on the surface of nanoparticle when they are in biological matrix is quite important for us to understand their behavior. So my group actually is an expert on analytical separation as well as spectroscopy and spectrometry measurements. Therefore, we actually develop open channel separation tools, including capillary electrophoresis to measure binding affinity, and also use flow field for fractionation to study the corona stability and formation. We also develop spectroscopy methods to study to screen a large number of interaction between nanoparticle and proteins and try to find out the fundamental of how the property of the nanoparticles or how the protein property would actually control or contribute to the interaction. Because for some of you who have rich experience in identifying protein corona, we know that we typically will find hundreds of proteins. And for most of the proteins, we probably cannot get a purified protein and then really study or confirm the interaction. So for our study, what we hope is that by studying the interaction between individual particles and proteins, so we can summarize some fundamentals that govern the interaction. For example, what kind of particle properties are most important for interaction, and what kind of property will induce what kind of like how strong and also how fast the interact because it's a thermodynamic and also kinetic issues, right? And also from protein part aspect, what kind of property of the proteins, like whether it's like just simple molecular weight, hydrophobicity, or just amino acid sequence. What kind of the property will control the interaction, so will contribute to interaction. So that to have some fundamental rules and then with these rules, then we can probably can have a better understanding of what are the proteins we found in the corona study could be of particular interest. So then to could contribute to their behavior in the system. So therefore, after developing this method to study its fundamental interaction, then we apply them to see to screen for the interaction between a large number of particles and also a large number of proteins. So I will actually introduce all this briefly today during my talk. So the first is actually this is the earliest work we did about protein nanoparticle interaction, because we were the first one to apply capillary electrophoresis to measure the binding affinity. Why this binding affinity is important? Because that actually give us quantitative give us quantitative comparison. And now it's not working. Oh, yeah, I touch it the same way, but sometimes it just go multiple slides. Okay. Oh. Yeah, I did that the same way. You see?
I think the keyboard has the issue here. It's
Okay. Okay. Yeah, because we believe the binding affinity can give us a quantitative measurement of just interaction strengths. So that when you have a variety of nanoparticles and then you test the affinity with certain proteins, then you can compare the interaction in between. So, and then to get some information about how the physical chemical property of the particle will contribute to the interaction itself. So, but this measurement is not trivial because for a lot of nanomaterials, actually they have a lot some optical properties that would interfere with the conventional measurement. So what we use is a separation-based technique. It's an open channel separation, so we don't have any gel or packing material inside the capillary column. Therefore, there's no interruption with the binding, which is a non-covalent interaction. So our model here is that we think when the protein binds to the nanoparticle, it will change either the size or the both size to charge ratio of the complex. So then we can see electrophoretic mobility change of the particle itself. And then using that electrophoretic mobility change, we can fit it in the Hugh equation to find out the binding affinity. So Kd is the macroscopic binding affinity here, and N is the cooperativity that's showing how the proteins on the same particle will affect each other's binding, whether it's a negative effect or a cooperative effect. So, okay, thank you. So what we do is that we incubate a nanoparticle in protein and then inject them into the capillary column and apply electric field to do the separation. So what we will see is two potential possible situations. One is that if the binding is has a very slow dissociation rate, which is slower than the migration time, then we could potentially see the protein bound nanoparticle peak versus the free nanoparticle peak. So then we compare the peak area, so then we can calculate the bound ratio. Another scenario is that the dissociation rate is very fast, so the complex is not stable during the separation process. So we have to put the protein into the separation buffer, so then we can observe the mobility shift of the nanoparticle when the protein is in presence of the buffer. So then the mobility shift of the nanoparticle then can be used to calculate the bound ratio, and then we can do the Hugh equation fit to find out the Kd level. So we apply this CE method to screen iron oxide interaction with a few proteins to identify surface functional group. Actually is very important to control in controlling protein interaction. So what we did here in this project is that we prepared two kinds of iron oxide particles, which they have identical size, and then the same iron oxide, iron 304 material, and it's super paramagnetic. So then their core size is around 10 nanometer, and then they are both coated originally with the polyacrylic acid. However, these two batches of particles went through different heating procedures. So one is going through a very long hydrothermal process, longer than around 12 hours, and the other going through a very short heating process, only lasts for 10 minutes. So for these two heating process, it actually generate two kinds of ligand on the iron oxide surface. One is we call it head group B, which from the short heating process, and this is still has the head group actually still has the hydroxyl group. But then for head group A, after the long-term hydrothermal process, it's already dehydrated and forming this five-member ester structure. So we call it head group A. So we confirm the ligand structure by mass spec and NMR. And what we see when we test the interaction of these two batches of particles with a group of proteins, we find these two batches of particles, although they have the same shape and very largely similar surface group, they still show different binding in strengths with different proteins. For example, most significant difference is observed with chromatography, so which actually more than 10 times difference in the affinity value we observe. And then the kinetics we observe in CE process is also quite different for some of the proteins. For example, for human serum albumin, for the one that carrying head group B, obviously it's a very fast dissociation rate during separation, and we can only observe mobility shift. But the one that heating for longer as long as 12 hours, they show very slow dissociation rate. So a very small head group differences already cause this kind of binding strengths and kinetic differences, which is a big surprise to us. So to confirm this kind of change differences, we also conduct mass spec analysis to try to find out what are the peptides locating at the interaction interface, trying to see why the functional group is so important. So what we do is that we, for this iron oxide surface, it has a rich carboxyl group. So then we do labeling, cross-linking between this carboxyl group and the amine group on a protein, and then we digest the protein, leaving only the peptides very close to the nanoparticle cross-linked onto the nanoparticle. So then after that digestion, what we found from the nanoparticle should be the peptide that locate at the interaction interface during this cross-linking process. So we believe those should be the peptides that very close to the nanoparticle when the protein binds. So we saw those peptides, and then especially in the chromatography, which I said we observe very large differences between in the binding strengths for these two batches of particle. We see actually several regions are affected. So what we think is that the reason that the surface group can be so important, although it's just the head group difference is observed, it affects the binding so largely is because there's a large interaction surface interface formed between chromatography and the nanoparticle. So then there is small change in the localized interaction strengths actually contribute to the overall affinity differences. Therefore, we asked our colleagues to help us to do computer simulation, which indeed find out that if we do docking, so for docking analysis, we use these two head groups as the docking ligands connecting to two monomers. And then for these docking ligands, they indeed show certain values in thermodynamic feature when they interact with the amino acid residues nearby. So if you look at this table, you could see the binding free energy towards the amino acid side chains around within these docking boxes are quite different between particles carrying head group A and particles carrying head group B. So indeed, the simulation supports our observation that even though it's just a small head group structure difference, it could induce big differences in binding strengths. So it is in particular for proteins that can open up and forming a relatively large interface with the particle. So then a subtle change on the surface ligand could affect the binding greatly. So the first surface property is so important, and then we'll try to see whether this will also carry on to the corona formation, because what we did previously is to study the individual binding. So therefore, we move on to compare particles that have different surface properties and how those properties will change the formation of corona, in particular the dynamics of the corona. So what do I mean by the dynamic feature of protein corona? Because we know that for protein absorption, it could be forming a hard corona, which actually very strongly attached to the particle with slow dissociation rate. And then there are also a soft corona layer, which actually has a very rapid exchange rate for those proteins in the soft corona with the proteins in the environment. So for this kind of soft corona, we believe is because the equilibrium can be established very rapidly, so they provide initial protection effect to the particle when they enter the biological system. However, the hard corona is the one that they could carry some of the proteins within the hard corona because they have very slow dissociation rate towards different environments. For example, they could be forming in the extracellular matrix and then being carried into a cell. So the relative components in the soft and hard corona is what we interested in finding how the surface of property of the particle will affect their formation. So we use the flow field for fractionation, which is also a commercially available separation system in our group. So this one, different than capillary electrophoresis, it uses a trapezoidal channel. And then this channel is kind of like a ribbon. It's very shallow, but and relatively wide. But similar like CE, it also has no packing inside the separation channel. So it's also one kind of open channel separation technique. Therefore, we believe it also have very low disruption impact on the formed corona on the particle, because we incubate the particle first in the biological matrix. For example, we use serum here, and then inject the mixture into the column, and then collect the particles at the end of the column. So for this material, it separates things by their size, hydrodynamic size. So for larger ones, because they diffuse slower than the smaller one, so then within the parabolic flow profile, inside the column, then those larger ones will locate at a relatively lower level within the parabolic flow profile, so then they migrate slower. But for smaller ones, they diffuse faster, so then their relative height within the parabolic flow profile is closer to the center, so then they have a faster flow rate. They move at a faster rate, so then they can come out earlier. So then eventually those particles will be separated by their size. So then when we have a protein that non-adsorb onto the particle, because this particle size is quite different than a regular protein, like with molecular weight around 100 to 150, and then they are much larger, right? So then we can separate the particle very well from the non-adsorbed protein, and when we collect the particle, those non-adsorbed protein will not be collected together. But we will have the hard corona coated outside of the particle when we collect the particle, because those proteins adsorb so stably, so inside the column they did not completely dissociate, or most of them still remain bound onto the particle. And then for the soft corona, this would be the protein that being depleted significantly in the column, but we should still find them if we use centrifugation. Because centrifugation for this nanoparticles, if we don't use the like very strong like outer centrifuge, just like a tabletop centrifugation device, then it would take a little bit longer time to sink down all the particles. So then during that time, both the soft and hard corona could be maintained. So then when we compare the proteins identified on the particle from centrifugation and also from the particle collected from our flow field for fractionation technique, then we will find out what are the ones that belong to soft and what are the ones that belong to hard. What we think is that if we can find a protein in both preparation, then those should be the hard corona protein. And if we can find the protein only in the centrifugation preparation, then those are the protein that belong to the soft corona. So to to prepare particles that with different coating, we also using just same iron oxide particles with the core size around 10 nanometer. But then this time the coating could be either polyacrylic acid, is very hydrophilic, a very dense carboxyl group on the surface, and also azelaic acid, which after the certain treatment, we actually remove half of the carbon chain. So then it has a smaller number of carboxyl group on the surface. And then we also buy some iron oxide particle that coating with the oleic and amphiphilic polymers. So this is kind of like the in between. So when polyacrylic is hydrophilic, azelaic is the most hydrophobic within this group, and then the amphiphilic has the in between hydrophobicity. So we want to see, although all of them carry carboxyl group on the surface, but then they have some hydrophobicity differences, and whether they would form different kind of corona on surface. And we also compare for the amphiphilic polymer coated particle, we compare the one that with a smaller core size and also the one that with a larger core size. So here again, summarize the property of these particles, and we see the zeta potential measurement show that they are negatively charged and with a relative comparable zeta potential, and then their hydrodynamic radius are also more than around like 15 to 20 nanometer, and then for the 25 they have around close to 30 nanometer. And the log p value is what I say is the index for the hydrophobicity, right? For the one that with a larger hydrophobicity, for azelaic acid, the log p value is 1.65, and then the PAA is the least hydrophobic, so then it has the smallest log p value, so which is 0.67. So we use the chemical structure to calculate this log p value. So then here, if we isolate the particle using our F4, which is a flow field flow fractionation technique, and also for the particles that isolate by centrifugation, we identified a soft protein on the surface. And this is after incubation with a depleted serum. So depleted serum meaning that we remove the serum albumin and also IgG using depletion column. But you see, because the albumin is so abundant in serum, we still see a lot of them in a binding. So if we identify both proteins in these two methods, the proteins in both methods, we consider this a stable bound protein, and it should be the hard corona portion. And then for the one that only identify in centrifugation preparation, we consider it as a like soft corona portion and with a very fast binding kinetics. So we confirmed that by also doing spectral counting, kind of like a semi-quantitative mass spec technique. So then we can compare the relative content for one particular protein in these two preparation. So indeed, for fast exchange proteins, which we also confirm it using capillary electrophoresis. So like I mentioned before, if you have a very fast exchange, then you will only observe mobility shift. But if you have a slow exchange, then you see the bound peak very clearly in CE. So for those 4 proteins, we actually confirm their binding kinetics in CE. And when we look at the relative protein content prepared by centrifugation, which is the DSC label, and then prepared by flow field for fractionation, which is labeled as DSF, then we could see for the one that with a very fast exchange rate, then you should see decrease in the percent peptides found in the F4 portion, which is the blue bar. For example, A1AT and then the ApoA1 protein showing fast kinetics. But then for transferrin and haptoglobin, then we could see higher ratio of the peptides found in the F4 method. That's because most of the proteins are being removed. The one that in the soft corona already being removed in F4. So the relative proportion of the hard corona will go up. So that's why we use the semi-quantitative method to confirm the that what we identify is actually the depleted. We could indeed identify both the hard and soft corona, and we can also do spectral counting to confirm some that not completely removed, but still have some remained in F4 portion, but then they have fast kinetics. So what we find out is that if the hydrophobicity of the protein particle surface increase, then the proportion of the soft corona also increase, which is the the light gray part of on this pie chart. So, and also when the size increase from 10 to 25 nanometer, we also see increase of the soft corona proportion. And another thing we find out is that when we calculate the surface hydrophobicity of the protein using the gravy method, so we use the gravy score to represent the surface hydrophobicity of some of the proteins we find in the corona. So we see that if the hydrophobicity increase on the particle surface, then also the hydrophobicity within the protein corona will increase, kind of like dissolve like, right? Or like to bind the surface that with similar property. So we see that again, the proportion of the hydrophobic protein prefer to bind to the hydrophobic surface, and also the hydrophobicity as well as the size of the particle will affect the dynamics of the corona formation. Then those kind of like when we identified, okay, the formation of the corona, then the next question is whether this corona indeed provide any protective or disruptive effect on the material when they interact with cells, for example. So therefore, we did a quick study of how this protein absorption actually could protect the cells from invasion of nanomaterial, which is the carbon nanotubes. So what we did is that we have the single wall carbon nanotube carrying carboxyl group on surface. So this carbon nanotube actually showed if they have some toxicity towards the human fibroblast cells. But if we coat them with the bovine serum albumin, then this toxicity would be decreased or even gone. So we want to see how this protein absorption will actually prevent formation of protein corona inside cells, so then could potentially provide some protective effect. So what we did is we prepared some carbon nanotube that coated by BSA, but using different mass ratio. For example, 1 gram of this carbon nanotube coated by 1 gram of BSA, or 1 microgram versus 1 microgram, and or 1 microgram of carbon nanotube coated by 4 microgram of BSA. So with a larger mass ratio, AFM shows that the coating on the carbon nanotube is thicker, right? And then this thicker coating actually will affect the toxicity. So when we incubate this with the fibroblast cells, so we see that coating with more BSA actually enhances the viability of the cells compared to the non-coated larger coating, more coating of BSA actually increase the viability, because we cannot distribute the bare carbon nanotube into serum. So that's why we did not include it in here. So and then also we quantify how much the surface coverage is, right? So we can observe it in AFM, forming seeing the carbon nanotube growing thicker. But here we isolate the carbon nanotube and quantify the protein absorbed onto particle, and also confirmed by measurement in supernatant. So then we see that actually for coverage surface of the CNT, if we have 1:4 ratio, then it's actually full coverage. But then for 1:1 ratio, there's still partial of the surface being exposed, a lot of surface being still exposed and not covered by BSA. So, and then we see how the uptake actually would be changed by the BSA coating. So when we have 1:1 ratio of the labeling, we see the uptake ratio actually higher than the one that with more BSA coating. So we think it could be the thicker coating prevents certain interaction with the cell membrane, so then reduce the uptake. Then we prepare the cytosol proteins to see how the cytosol protein actually can compete this BSA coating off, because it's not cross-linked, it's just a non-covalent binding, right? So it's a absorption of the albumin onto the tube. So then in the cellular environment, because albumin concentration will be much lower, so then it will definitely be replaced by the cytosol proteins. But we see that with a thicker coating, then of course less displacement actually will happen. And then for the one that was So all these numbers, meaning that the first number is the tube mass, and the second number is BSA mass, and the last number is the cytosol protein mass. So we see that actually with more cytosol proteins, a lot of BSA would be depleted from or displaced from the surface and being replaced by the cytosol proteins. And we further identify those proteins and compare the total number of proteins identified in the carbon nanotube that only interact with directly with cytosol protein, or the BSA coated nanotube at the 1:1 coating ratio with one mass ratio of the cytosol protein, or the 1:4 coating tube with the same mass of the cytosol protein. So the blue bar represent the one that actually shared by all three preparation. Those seems to be the very strong binding, probably still just attached onto the carbon nanotube anyway. It doesn't matter whether it has a BSA or no BSA on the surface. And we also find some unique protein which is labeled in yellow bar that in this three preparation. So for example, for the one that directly interact with cytosol protein, we see they absorb cholesterol, which is actually important for endocytosis, and also for some proteins that directly related to apoptosis, which we believe could be some of the reasons that lead to low cell viability if we don't have a sufficient BSA coating. So this kind of this is and then we see also try to analyze the physical chemical property of the proteins, trying to analyze their gravity, the hydrophobicity, molecular weight, and pi. But the changes are not very large, so therefore we don't think these three like very easily accessible general protein property actually would tell us much information about what are the proteins that could be absorbed, or what are the key feature of the proteins that determine absorption. So anyway, for this study, we think that if we have a certain protein layer on the surface, which is very can potentially provide some protection effect to reduce cell cytotoxicity of some of the nanomaterial, and this is because the absorption actually prevent absorption of cellular protein on the surface. But like I said, all this corona study actually give us a large number of proteins to kind of speculate their potential function. But still, for fundamental understanding of what property of either the protein or the nanoparticle control the absorption is still very hard to derive that kind of information. For example, if we analyze the overall property of the protein by molecular weight, pi, or hydrophobicity, we didn't get much useful information. So what we want to see is now if we have like a model proteins and also model particles, and then we want to see their interaction and how we can see in a high throughput manner. So CE is nice. You can get very quantitative measurement for the binding affinity, but it's each injection only one sample, right? So if you want to screen tens of particles or tens of proteins, you need to run tens or even multiple times tens runs, right? So that's very slow. So we would like to use spectroscopy screening. Then for optical signal, you can couple with multi-well plate, then you can scan a lot of samples at the same time. So what we developed then is like using a furosemide molecule, which is non-fluorescent molecule, until it reacts with the primary amines on protein. So we know that protein has a lot of lysine or arginine. Those are primary amines so that they can be labeled by furosemide. So this molecule is very nice that it reacts very rapidly, but if it would not react with primary amine, then it will quickly hydrolyze into a non-fluorescent and non-reactive molecule. So then this molecule only label the surface amine on protein and will not have access into the amine that embedded inside the protein during its folding. So we think there could be two very simple scenarios. So the particle binding could block the surface amine, so prevent them being labeled, or it could open up the protein and leave more lysine exposed and accessible to this dye, so then it could have more fluorescence signal read out. So therefore, we can then we actually study the interaction of a group of proteins with quite a number of particles. They could be polystyrene particles with a diameter around 40 or diameter around 80 nanometer, and silica particles that we control the amination process. So that means control the surface carboxyl group on the density. And then we also buy this iron oxide nanoparticle that coated by PEG or amphiphilic coating to see how the surface and core properties will affect their binding. And for this study, we choose only a few proteins to initiate that. And then these proteins also we want to choose to represent a wide range of molecular weight and wide range of PI. And here is what we confirm, that the labeling is actually correlated to the number of lysines group of each protein. So it's proportional. The fluorescence signal actually is proportional to the number of lysine on each of the proteins, so then only target the surface protein. And this is the fluorescence profile we obtained for the 3 polystyrene particles that we tested and around all these 7 proteins. But we see majority actually showing increase of the fluorescence, which indicates certain kind of conformational change and open up more lysines to be labeled. And if we're using the fluorescence profiles for all particles, and here we have a total of 9 types of particles, and we see the fluorescence profile actually can group the particles very nicely by their core materials and also by their sizes. So if you look at the green signs, those are the polystyrene particles. And the triangle green and the green circles are the 2 polystyrene particles with very close core diameter, like 42 and 43 nanometer. But then the square, the diamond one, actually is the 85 nanometer polystyrene particle, and then they are very well separated after we do principal component analysis of the fluorescence profiles, and then they're being grouped separately. And then the blue signs are for the silica particles, and then if we enlarge that area, we could see they are also being grouped by the process of the amination. And then the iron oxides obviously very nicely grouped by their surface property and also small change even in the size. So, and then we expand this screening to a larger number of proteins, and then hopefully again here we want to have a wider coverage of molecular weight, PI, and hydrophobicity. And here a total we screen 21 proteins, and some of the signals are shown here. And again, for polystyrene particles, we could see enhancement in the fluorescence label, and in particular for the smaller one, we could see more fluorescence enhancement compared to the smaller one. But then for silica particle, we didn't see much very significant enhancement. So one meaning is that compared to the protein by itself, if the labeling occurred to the protein by itself, it didn't change much, right? And if it's the ratio is larger than 1, that means after incubation with particles, we see more labeling. But if the ratio is smaller than 1, then that means after the interaction, we see less labeling, which could be the blocking issue. But the change overall in silica particles is not as large as the polystyrene particle. So then we went on to study more about the interaction, the protein that's showing high signal change when they bind to polystyrene particles. Because like I mentioned, so a lot of this change could be due to the conformational change of the protein. So that means our method not only can tell us, okay, initially what proteins could interact with each particle, but also could tell us what proteins may experience larger conformational change when during the interaction. And here we are showing that the fluorescence signal change indeed can also tell us a certain binding could be relevant to the binding affinity. So when we do curve fitting, we see different dissociation coefficient. So this is kind of like another way to approach the affinity, even using the labeling method. But more importantly, we want to see whether the particle property also still dependent on the interact, will also be a key determinant of the interaction. So when we no longer control the surface amine density, now these 2 batches of silica, although they have same core size, 50 to 80 nanometer, they're no longer being separable. But for the polystyrene particle, we still see this 48 and 85 the sizes have quite significant difference in protein interaction. But if we use the fluorescence profile or using the particle as the variable to group the proteins, like I said, it's very hard to find a particular feature or the feature that we are very familiar with for the protein to see, okay, whether it's a larger protein that binds stronger or it's a more positive protein that binds stronger. We didn't see that kind of relationship. However, if we do this k-means clustering method, then we reveal actually two groups of proteins. One group of protein which show very high fluorescence change, and which is included in the red kind of like a triangle or irregular shape though. So all those proteins show very large change in fluorescence. And then they are very interestingly could be some with high structural flexibility. For example, catalase and beta casein. BC is beta casein. And also they could be heme-containing proteins like hemoglobin, catalase, and CC, which is the cytochrome C. And transferrin and coalbumin actually has very high similarity in their structure. And then they also could be for transferrin and also hemoglobin, they actually could form oligomers like tetramers. So like high structurability as well as heme-containing proteins showing high fluorescence change, which could potentially due to conformational change, right? Because if they already have structural flexibility, and then once upon binding to particle, the change could be more significant compared to, for example, protein like HSA, which already being flexible. So then the differences between binding or no binding to particle would not be that large compared to the protein that a lot of areas are a little bit rigid, but some of the loop structure, which highlight in orange colored, are the highly flexible portion. And also, like I said, cytochrome C, although it's rigid, it can form, it has a heme group. So those are also another feature, that common feature that we find in the group of protein that showing high fluorescence change. And what we think is that the labeling could be due to conformational change differences, because those will open up more license to be labeled. But more importantly, we want to confirm that, and so we use partial protease digestion. So this protease digestion is very quick. We only conduct 5 minutes digestion with trypsin, and then we see if the protein is incubated with the particle, we see actually some of the proteins is showing high fluorescence change, indeed being digested much more rapidly than the protein that's showing very low fluorescence change. For example, the first column, I think they're missing some. Could you, yeah. So the first batch of the gel, 3 column, is the human serum albumin, which show very low fluorescence change. But the catalase, CAT, show, and also transferrin, both show very high fluorescence change. And then they show very significant digestion even during this short protease incubation time, indicating that after binding, particular to polystyrene 40A, polystyrene particle with the core size around 40A, it actually changed, the conformation of the protein may have changed that facilitate protease digestion. So open up more regions for the protease to digest it. But silica nanoparticle, which show very low fluorescence labeling change, it actually does not affect the digestion at all. So here we remove the effect on the particle to trypsin by pre-incubating the proteins with particle. So then we also incubate the proteins with particle at a high protein coverage, so then to prevent impact from particle itself to trypsin. But if the trypsin activity is affected by the particle itself, then the HSA effect should not be the same, right? So then that's what we believe is actually due to binding induced conformational change. And here is that if we have other proteins in the mixture, because they compete for the same binding site on the particle, it actually could alleviate this susceptibility to protease digestion. So due to competitive binding, so kind of again agree with the protection model. So if you have other proteins to protect the surface and prevent the binding to certain proteins, then the conformational change of those proteins would not be observed anymore. And the binding conformation also could be related to particle aggregation. And here we actually have not done very systematic investigation yet, so this is just a very preliminary data. I don't want to spend too much time on it. But we further applied partial digestion and then try to identify what are the peptides that are being digested at the first very initial, very quick digestion round, which we believe will be the peptides being affected during this conformational change, and then compared to the protein that remain bound, but then the digestion takes a much longer time. So what we did is that we did the partial digestion, isolate the peptide first, and then for the remain bound protein, we continue to digest them and then identify it in a mass spec and compare the cutting site in both the filtrate and then in the protein that remain bound onto the particle. So what we see is that, so for the peptides in the transferrin and also catalase, which we see more effect from or higher fluorescence change and also a very rapid protease digestion, then we indeed identify more peptides in the filtrate compared to HSA when they incubate with polystyrene particle. And if we analyze the cutting site that being affected by protein binding by comparing the cutting sites identified for the protein remain bound on the particle and then the protein in the filtrate, and then illustrate the position of those peptides on transferrin, we see those are the structure that actually labeled as the high B factor, which has a high flexibility, so being affected. And for catalase, it's very interestingly, we find that the cutting sites that change a lot actually very close to the heme center, so which is the activity center. So then when we move on to study the activity change, indeed we see catalase activity is being significantly reduced after it binds to polystyrene particle with the diameter around 48. So the top picture showing the foam test that showing the oxygen generation catalyzed by CAT catalase, and then so we use that the foam generation to illustrate the activity of the enzyme and showing the enzyme activity reduction after binding to the particle, and which we believe is because the binding site is very close to the activity center. And then we suspect the heme group, why all of the heme containing proteins we find actually showing very large fluorescence change is because the heme group has the iron. And the iron could possibly kind of like a metal bridge acting between the protein and then the aromatic polystyrene particles, and then which once the heme group actually is changed, it will change the lysine hydrogen bonding with the peripheral amino acid side chain that will open up the protein structure and enhance more label. So similar to protease digestion, the activity decrease can be reversed by the presence of other proteins. So that means if, so here I want to show that the fluorescence labeling could potentially reveal protein conformational change upon interaction with nanoparticle. And also for this kind of change, when we study individual particle and protein interaction, we try to discover the relationship between the property and how those properties will affect the affinity, the binding. But we also want to consider that when other proteins being present, then how they will change the interaction. So, and therefore we want to apply the partial digestion procedure. So hopefully this method can be continuous to develop, and so that we can look at a more complex mixture. So how the binding occurred in a complex mixture without isolating them. So at the end, I would like to thank my group and also thank you for your attention, and I will be happy to answer any questions that you have.
Thank you, Wenwan. Very interesting presentation. Now we'll open the discussion, questions from the audience, and then we'll follow with questions from people on online. So any questions from the audience?
Thank you. It was a very interesting talk. One question that I have is when these nanoparticles are taken up by the cells, they encounter a variety of pH coming from high acidic pH of 4 to sometimes extremely alkaline up to 11. So do you also can you also implement these pH in your study and see that how it affects these hard protein corona and these protein specific bindings?
Yeah, for pH, we actually did a very brief test with the fluorescence labeling. We actually compare labeling at 7.4 and more basic condition to see how the pH actually affect the folding. But for a lower pH, this may not be the a right way. So that's why we also continue to develop this partial digestion method to try to look at that in a quicker manner. Because for low pH, it will affect the lysine labeling activity. So it will be more active when we have like a pH above 6 because the lysine group is deprotonated.
More questions from the audience? I have a more philosophical question because we all use in vitro cellular assays for screening of nanomaterials, and what you show here is the protein coronas will be completely different if you have this artificially put in there proteins, which they will affect the protein characteristics on the particles, and then that will affect the bioactivity, the uptake of the particles. So, I mean, from everything you have shown, it makes me rethink that maybe cellular screening is not the most appropriate way of screening nanomaterial interactions at the cellular level.
I think it should still be very valid to do it that way. But what I just when I also talk to your people, I try to explain that we study the fundamentals and we want to get some rules that we try to understand more. So then when you do the screening and then you see, okay, there are a group of protein that could be bound. So then we apply this to explain, okay, to see, okay, whether this are indeed potentially highly interactive proteins to confirm that. And I think it's also very important to see it in vivo. So like for any of the in vitro study, then we isolated them. It could not be the same way as what they experience in. So then.
But we do know that there will be completely different proteins from the in vivo physiologically relevant. So it makes me rethink. I mean, I agree with you that we really need to at least include in our characterization of these systems the protein coronas, which is very rarely is done for cellular studies. So at the minimum, I think we should really include as part of the characterization metrics this protein characterization. So at least we know what is going to end up on the particles, which may not be the physiologically relevant ones, but at least we know what's in there and what would drive the certain activities.
Yeah. Yeah, but also I think it's very important that actually your group also started to do that, to collect different portions, like from the track of exposure, and then during the travel inside the body, animal body, so which part being changed in the corona formation.
Okay. Yeah, one more question from the audience, and then we'll switch to have questions from the.
It's a very interesting talk. So my question is about when you put different proteins on a nanomaterial, right? These proteins are in a conformation and a distance they never really experience physiologically. So do you have a method to study nanomaterial corona protein-protein interactions?
So you mean like a protein interaction with a particle, and then that protein interact with the following protein?
With other proteins, because the particle forces proteins together in a distance very closely to each other.
Yeah.
Do you have a technique to further analyze that interaction?
That we have not. That's why we get this partial digestion and also the gel results. So that actually our first attempt to analysis the mixture effect, because gel you can separate the proteins by their molecular weight. So then you could potentially see how those interaction will, but we indeed notice this so-called crowding effect. So then actually concentration or condense on the particle surface could potentially induce more protein-protein interaction. So that's what I expect is happening during the aggregation phenomenon we observe. So when you increase the protein concentration, because originally the protein bind, for example, transferrin, and we show some conformational change, and it could open up certain hydrophobic region and induce more, or like I said, for proteins that can form oligomers, then when they absorb onto the protein, onto the particle, then they may also form more oligomers, and then to that kind of interaction. So initially we try to use gel to assess that.
I just have a comment on the previous question. We did develop in the past a method that you can actually use the particle-particle interaction when they're coated with proteins. We use the AFM and we put the particles actually on the AFM tip and on a substrate, and then we put this in biological media so the proteins attached, and then we could actually measure the interaction force. We could push the particles together and then we pull them apart so we can see the adhesion force. Sometimes the repulsion, they don't adhere at all. So, and we saw that it's very much dependent on the proteins you have and the particle size and the particle material as well.
Yeah.
Yes, we didn't characterize the corona, yeah.
So everything that you have done over here is pretty much based on digestion in vitro, essentially in a test tube. If this particles with corona were to get inside a phagocytic compartment, you will have proteases present over there. What kind of digestion will you see inside a phagocytic compartment? Would it be very similar to what you are seeing outside, or will it leave any kind of peptide on the surface of the particle, or will it completely digest those peptides totally?
That's an interesting question. We have never looked at particles that are already in the endosome, which where you said, or also lysosome, where you said the high level of protease percent. Yeah. But also the pH will change, and the pH change could change the corona. Yeah. And if they release, then definitely they will be digested easily.
Okay, I think we have to stop here, unfortunately. Thank you, Wenwan, with a very interesting presentation. I'm sure we got out of this with many, many more questions that coming here today, but that's the complexity of these issues. And I'm glad that you're taking the lead on this and trying to figure out what's happening. So thank you all.